Good morning, everyone, and welcome to Wheaton Schroeder's thesis defense. This is definitely a special day for me because this would be the first thesis defense from my group. Wheaton hails from Cedar Falls, Northern Iowa, and he went to Iowa State to get his bachelor's, double majors in mathematics and chemical engineering. Then Wheaton moved here and started working with someone else in the department. When I met Wheaton first, I think maybe on my second or third day when I moved to Lincoln in a game of cricket. So how many white dudes you see like in the US plays or loves the game of cricket? So yeah, that's the first time I met him. And there was a time when I know that given his background, he was working in an experimental lab, and I was always wondering, what is he doing in that experimental lab? Anyways, things moved differently, and in 2016 December, he then approached me because at that point he was looking for a new lab, and at that point I was about to recruit another student from a different country, and that student could not make it. A position opened up, and then I said, hey, why don't you come and work with me? And things started from there. So today Wheaton will present all the work that he did over the last 4.5 years of time, and I will leave it up to him to talk about what he would like to do next. Wheaton, take it away.
Yeah, so thank you for that kind introduction. As he already said, my name is Wheaton Schroeder. I'm a 6th-year PhD student here, but I've been working with Dr. Saha for the past 4.5 years. And today I would like to talk to you about the work that I've done under him. Really the key to this work is these 5 highlighted words, tools for genome-scale models, because that's really at the heart of both my thesis and dissertation, as well as this presentation. So there we go. So I'd like to point out at this time, I have 5 publications that are currently in print. They're all first author, as well as a further 3 first author publications undergoing peer review, and a 9th publication that we hope to finish before I leave UNL. At this time, I'd like to mention that just due to the scope and the time limits of this talk, I won't be able to talk about everything here, but I'll give you some QR codes and links at the end of this presentation so that you can follow up and satiate your curiosity about my other works. So I'd like to start by giving a bit of background about the field of systems biology and the kind of models that we use. Then I would like to talk about the first model that I developed, which is a model for a pigmented fungus, and how this led me down the research path of building tools for genome-scale models. Then I would like to talk about a dynamic whole plant model that we used. The first one used one tool, the second one used another tool. And then finally, I'd like to talk about a lot of the work that I've done since my comprehensive exam, which has been focused on building mathematical tools for synthetic biology. Finally, I'd like to talk about my research going forward, both the research that I've done here at the Saha Lab, as well as my own personal research career. So to begin with, let's give a bit of background and overview. So the kind of models that we work with are called stoichiometric models. They are a set of reactions which occur in an organism, and we'll define that in what is called a stoichiometric matrix, where we give stoichiometric coefficients in this matrix between metabolites and reactions to establish linear relations between the two. These are built from the genome, so what we're going to do is look at the genome of the organism and try to determine what reactions would be supported by that genomic information. We would call this stoichiometric model a genome-scale model, or a GSM, if it has all genome-supported reactions. So we build these through establishing gene-protein reaction, or GPR links. Sort of due to the availability of genomic information and sort of the comparative ease of reconstruction, most of the genome-scale models, about 94%, are for prokaryotic organisms at this time. Stoichiometric models are not just the stoichiometric matrix, however. We will also provide lower and upper bounds, as well as sometimes reasonable physiological bounds for these models. And these are defined from various factors such as thermodynamics, in vivo experimental data, and sometimes modeling necessity. Once we have created this genome scale model of metabolism, this allows us to perform a systems level analysis of metabolism. So what a systems level analysis does is it allows us to use computational tools to sort of look at the whole of metabolism in one go. So this allows us to do various things, such as predict gene knockouts, which will create a valuable bioproduct, or be able to change gene expression or predict changes in gene expression toward the same end. Finally, we can also discover something new about the organism that we didn't know was there by trying to reconcile our model and experimental data. So overall, taking a systems biology first approach can help us save time, labor, and grant money in the long run. So in acknowledgement of how useful these tools have been, there are various analysis techniques which you can apply to them. The most basic is called flux balance analysis, or FBA, and this applies the pseudo steady state assumption, basically the assumption that we're looking at such a small slice of time that the levels of enzyme and gene expression will not change. So we can assume that our concentration is also not changing. And this will allow us to take what I call a picture or a snapshot of metabolism at a single point in time. A similar tool to this is called dynamic flux balance analysis. This drops our pseudo steady state assumption and allows us to look at both reaction rates and metabolite concentration across time. And so I like to use the analogy here that this is more like taking a video rather than taking a snapshot of metabolism. So one of the tools that I'll be talking about today will actually offer a new approach to dynamic flux balance analysis. Now, when we go to reconstruct an organism, our knowledge of that organism is never perfect. Therefore, in our initial reconstructions, we're always going to have gaps. So the state-of-the-art tools for addressing these gaps are called gap find and gap fill. And sort of the first tool that I developed and the first tool that I'll be talking about is a new gap filling approach that works better for the organism that we're going to talk about in a minute. Finally, there's also tools which take a systems biology approach to designing systems biology approach to designing synthetic biology interventions. So just a quick definition, synthetic biology is the repurposing or engineering of an organism to perform some kind of new function. So this OptCircuit tool is sort of the current state-of-the-art tool, which allows us to predict different different synthetic biology applications. So I'll begin by talking about modeling a pigmented fungus, which led me to my first genome scale modeling tool. So the pigmented fungus in question is called Exophyella dermatitidis. I'll just call it Exophyella from here out because that's a bit of a mouthful. Exophyella is unique in that it produces two different pigments for its defense. The first pigment is called melanins, and these are deposited in the cell wall. The second pigment is called carotenoids, and these are deposited in the cell membrane. And these two pigments give it a polyextremotolerant characteristic, which means it can survive in many extreme environments, whether that be pH, toxicity, or radiation. And this is quite interesting for us because Exophyella does this with a very small genome and a very small proteome, especially compared to other eukaryotic model organisms. Another thing that interested us about this particular organism is that the key enzyme in melanin production, both in this organism and in humans, is called tyrosinase. And we find that the active site is very well preserved across a wide range of species. So this makes us question if we could possibly use this as a model system for melanin production. We also have an in vivo research collaborator who has panels of different mutants of Exophyella so that he can test some of our model predictions in vivo. So our research questions really came down to two big ones. The first is, what is the cost of these defensive pigments to the organism? And the second is, can Exophyella serve as a model organism both for polyextremotolerant systems and for melanin-producing systems. So to answer these research questions, we took the approach of using genome-scale modeling. So I'm just going to quickly step you through the process of genome-scale modeling. So we begin by consulting public databases to build a list of reactions which we believe are supported by the genome. A few example databases are shown here. We then make sure that these databases are balanced both in charge and elements. And then we will construct what's called a biomass equation. So a biomass equation is a pseudo-reaction which allows us to simulate the growth of the organism by consuming biological building blocks like amino acids and lipids. We'll then assemble our biomass equation along with all the other reactions that we have into a genome scale model of metabolism and apply various analysis techniques to try to reconcile this model with our in vivo data. Now, this is where we actually ran into problems in studying Exophylla because we noticed that Exophylla was very understudied, and therefore we had a lot of gaps in our knowledge of how this organism works in terms of metabolism and what its genome encodes. So this meant that we had to go to a gap filling technique. So the traditional gap filling technique is to consult these same databases that we did before. And once we have built our incomplete model reconstruction, we will define a database of reactions that we think can solve these gaps. Then we could apply state-of-the-art gap filling techniques like gapfill and GenDev using this model and database to get a per metabolite solution to these particular issues. So for fixing metabolite A, we might have reactions A or B that need to be added. Now, this becomes problematic when you have an understudied organism like Exophylla, because what happens is we'll have hundreds, perhaps even 1,000 metabolites to fix, and doing this all manually causes several different issues. The first is that it's obviously very time-consuming, and that it's dependent on the curator, depending on what, for lack of a better weight or priority you give to fixing the various gaps. We can also get something called thermodynamically infeasible cycles. This is where reactions just sort of are modeled to occur at any rates that is outside of what is biologically feasible because of the structure of the model. And I can get into that later if you have questions about that after the presentation. So we had a lot of these issues when we went to address this for an understudied organism. So we created a new approach to gap filling called OptFill. The first step is going to be the tick binding problem, which is going to identify all these thermodynamically infeasible cycles that could occur between the model and the database. Then we will feed those results to what's called the connecting problems, and it will try to solve on a whole model basis how to fill as many of these gaps as possible while minimizing the number of new reactions added. So we call this both a holistic and a conservative approach because this whole model and we're trying to minimize our changes. So this allows us to overcome several limitations of the current state of the art. The first is we can avoid these ticks now. And from a small modification of the tick finding problem, we can actually identify inherent ticks. So we get almost 2 different genome scale modeling tools for the price of 1. We can also guarantee that we're adding a minimum number of new functionalities. And finally, we're minimizing our dependence on who's doing the model building because the results are for a whole model as opposed to per gap. So you just have to pick one result and run with it. So in our particular organism, now we knew that we needed to build a set of databases in order to apply our OptFill tool. So we looked to a related genus called Aspergillus, which is a melanized fungus. It does not produce carotenoids, but we saw that there were 4 different genome scale models for this organism that had already been produced. So we looked at the overlap in functionalities between these different organisms, and we concluded that those functions that are a complete overlap between all 4 models would represent core functions to melanize fungal metabolism. We then took some of the agreement points, such as 3 out of 4 agreements, 2 out of 4, and 1 out of 4, to make a sequence of 3 different databases in order to apply and gap fill our particular model. So using our reconstruction that we built from our knowledge already and the gaps that we filled both by adding the core functions as well as defining databases, along with various publicly available databases. We applied OptFill 3 times to our incomplete reconstruction, using each of those sort of agreement databases sequentially. And this allowed us to produce our final model, which is called IEDE 2091. It accounts for about 1,700 reactions, about 1,900 metabolites, and about 2,100 genes. And now that we've recreated our model with this new genome-scale modeling tool, now we can begin to answer our research questions. So if you recall, our first research question is, what is the cost of these defensive pigments to the organism? To do this, we use a concept called shadow price. So shadow price is going to be the change in objective function for producing one more unit of something. And in this case, that something will be a defensive pigment, such as carotenoids or melanins. Generally, you'll encounter the concept of shadow price in economics, which is defined as the estimated price of a good or service for which no market exists. So this little cartoon here sort of gives an overview of it. So the economist says that early delivery comes with a shadow price of $10,000 because it produces a better relationship with the customer. So then the management will pay up to $10,000 to cash in on that very real value. So that's sort of how shadow price works in a biological context as well. So when we apply shadow price, we have 2 major results and 2 major hypotheses coming therefrom. So the first is that we found that carotenoids are always more expensive than melanins, even on a per atom basis. And we found this rather odd because, as I said before, the melanins are going to be in the cell wall and will be the first line of defense. The carotenoids will be the secondary line of defense in the cell membrane. And there's no function that we know of that that carotenoids can perform that melanins cannot also. So this led us to the hypothesis that carotenoids have an as yet undiscovered role in exophylla metabolism. Our second result is that each pigment's price varies based on the nutritional source that we provide the organism. And this allows us to hypothesize that exophylla produces so many different defensive pigments so that it can minimize its overall pigment profile cost. So that was our first research question. Our second research question looked at how similar or how much of a model organism could exophylla be. I won't be able to go over all the results here due to time limitations, but I particularly wanted to take a look at this particular image. So this shows the amino acid sequences for the active site of tyrosinase, which is copper binding domain A and B. Again, tyrosinase is the key enzyme in melanin production. And we can see that the functional amino acid residues, such as noted here by a letter number in colored box, denoting its function, are very well preserved between humans, which are going to be these top 5 rows, and exophylla, which will be these bottom 4 rows. Another thing that we can also see here is that residues whose mutation leads to what is called oculocutaneous albinism type 1 are sometimes preserved. These mutation sites are shown in these white boxes here. So we can see that this particular residue is very well conserved between the 2 species, humans and exophylla. So this would allow us to hopefully explore medical applications for this work in future. So this first project focused on FBA. The second project is going to actually focus on the next tool that I talked about in the introduction, which is dynamic flux balance analysis. And we're going to apply this to study the life cycle of a particular plant. So again, dynamic flux balance analysis is like taking a video of our metabolism across time. It has a few advantages compared to FBA, including that it can have greater accuracy. You can model time dependence, and you can better model complex systems. However, it has a few limitations because it can be more complex to implement, especially since it's a large system of ordinary differential equations. It can also have issues of numerical stability as well as numerical tractability. In the past 2 decades, there's almost exclusively been 2 approaches used to perform the FBA. The first is called the static optimization approach, or SOA. It solves in a cumulative and step-by-step manner and is easy to implement and utilizes linear programming. However, its limitations are that it uses a first-order Taylor series approximation, which limits both solution stability and solution accuracy when you go to solve, especially for a large number of time points. So as we were trying to model the full life cycle of a plant, we found this too restrictive and too limited in order to meet our needs. The other approach is the dynamic optimization approach, or DOA. It solves for all time points simultaneously and is accurate and has stable solutions. However, it utilizes nonlinear programming, which can give us problems when it comes to computational cost and time, and it can also be quite difficult to implement. So if we're, again, looking at a large model and a large number of time points to solve, this is problematic to implement as well. So we created a new approach to dynamic flux balance analysis called the Optimization and Explicit Runge-Kutta-based approach, or ORCA, which we believe sort of meets in the middle of these two extremes. So it has several features similar to the SOA in that it solves in a step-by-step manner. Each solution builds upon the previous, so it is cumulative, and it utilizes linear programming. However, it's improved because it lowers our error floor. It gives us higher accuracy and stability simply by using a Runge-Kutta method. And we're able to make use of multiple different Runge-Kutta methods because this is generalized enough so long as we meet certain criteria. Now that we have built our tool, we're going to take a look at the plant that we're going to study in this particular case. So the plant that we chose to study is called Arabidopsis thaliana, or I'll call it Arabidopsis for the rest of the presentation. And we chose to study this particular organism because it is, since the genomics revolution, our model plant system. So if you want to do anything in a plant, you generally start in Arabidopsis, and then you'll work to more complex plant systems. So even though the in vivo plant has a lot of different tissue types and is somewhat complex, we sort of simplified this in our model to 4 representative tissues: a leaf, stem, seed, and root tissue, with various interactions both between the tissue and across the system boundary shown by this dashed line. Now, we decided to model Arabidopsis just for the core metabolism to show the usefulness of the ORCA approach to DFVA rather than create a genome scale model. So the leaf is about 500 reactions and functions for gas exchange, photosynthesis, and as a source tissue. The root has about 150 reactions and functions for exchange with soil, as well as nitrogen reduction, respiration, and transport. The seed model functions for storage of metabolites, as well as a way to model the cost of reproduction. And finally, the stem model exists to logically link everything together, as well as be a source for transpiration, respiration, and storage. Together, we're going to call this the Path 773 model for the 773 unique genes represented in this model. So we then applied the ORCA to DFVA to the Path 773 model, and we did this modeling 7 distinct life cycle stages. So this includes seed germination, leaf development, flower production, and silique ripening, which starts to include senescence, as well as transition steps between those 4. We constrained the macro scale behavior of this particular model so that we would meet various sort of nutrient flux checkpoints as well as biomass checkpoints so that we can recreate in our model what we would see in a lab. This allowed us to leave the micro scale behavior unconstrained and allowed us to investigate 2 metabolisms that we were particularly interested in. The first was sulfur metabolism, because that is generally quite understudied, especially compared to nitrogen metabolism, which is another important micronutrient for plants. We also wanted to study the flow of water and how metabolism influences that across time. So this is an example of our whole plant sulfur metabolism when we're looking at reaction rate across time. So sort of as expected for sulfur metabolism, we're going to uptake sulfur from the root as a micronutrient. Much of it is going to be processed in the leaf to create biomass as well as various important amino acids. And then those will get transported to the seed in order to construct the seed tissue. And so the direction was largely as expected. However, we noticed that in the leaf, particularly, we had a pool of metabolites be produced for sulfur-containing amino acids, which indirectly facilitated the transport of other metabolites. For water flow, we made a similar diagram, but I'll just share the broad results here. First, we were able to show the strong diurnal patterns of water flow across the plant that are caused by transpiration and metabolism. We were also able to show in this model, which was a slightly surprising result, that the contribution of water from the metabolism to that which is transpirated is actually quite significant. So that's something that we did not expect to see that we did. So that basically covers all my work up to my comprehensive examination, which was last year. So now I'm going to talk about a little bit more in depth some of the work that I've been doing since then. And it all focuses around computational tools for synthetic biology. So as I said, synthetic biology is the redesigning or engineering of an organism for a new purpose. As with any engineering application, we're going to have three distinct steps. The first is going to be design, then build, then test. So here we're more concerned with the design stage because this is where systems biology and computational approaches can really shine. So we decided to consider a concept for this called phytoremediation, where we use plants to remove, transfer, destroy, or stabilize soil contaminants. And as with the Orca to DFDA, we're going to start in Arabidopsis because that's our model plant system. So we decided to start by simply designing an Arabidopsis system which could respond to three different heavy metal ions, which might be present as a result of, for instance, pollution from mining. So to accompany this concept, we had to reconstruct a library of bioparts, which included promoters, transcripts, terminators, and enzymes that we think might be able to allow us to create our different logic gates, as well as create the desired behavior that we want. Using our concept in our bioparts library, this is where our first tool, or where the first tool I developed came in, which is called the Eukaryotic Genetic Circuit Design Tool, short UGene-SID, which tries to put together triads of promoters, transcripts, and terminators, which produce the desired synthetic biology logic. We then will pass our design to the Eukaryotic Genetic Circuit Modeling Tool, or UGene-SIM, which will perform a dynamic model of our design results in order to screen for potential bad behavior or undesired behavior. And then we can repeat this however many times we want until we have suitably explored our solution space. Then we'll be able to screen our design solutions for those that we think are going to be best built in the organism in question, and then finally select a set of solutions to build in the organism. So our bioparts library, like I said before, will consist of promoters, transcripts, terminators, and proteins. And each of these will have certain characterizations associated with them that are just sort of quantitative characterizations of qualitative traits. So this is sort of how the tool works. So for promoters, we'll have a normal state amount, some kind of definition of what the promoter strength is relative to other promoters in the library, as well as a promoter leakiness factor, for example, so that we can account for problems like leakiness in a promoter. So once we had built our once we had built our bioparts library and had devised some of our conceptualizations, we defined our conceptualizations as a logic table because this is a circuit design tool. So this is an AND logic table where our inputs are going to be the presence or absence of copper and the presence of or absence of zinc, indicated by a binary 1 or 0. And then the output of this system is going to be the presence or absence of green fluorescent protein at a high enough level to show the phenotype. So this is going to be shown again by 0 or 1s. And when we started with our bioparts library and started using these conceptualizations, we quickly ran into a problem called bistable orthogonal designs, or BODs. So bistable orthogonal designs have two distinct characteristics. The first is that the circuit will have two stable states. So for instance, the first stable state is going to be when TET-R and GFP are produced, and TET-R is turning off the expression of CI. The second distinct stable state will be when CI is produced and turning off the expression of both TET-R and GFP. So it's bistable. The second characteristic of these solutions is that they're orthogonal. So they're unresponsive to the circuit inputs that we want the circuit to respond to. So as we can see in this particular design, there was no responding biopart to these particular heavy metal ions. And then what happened, at least in our early stages of developing this tool, is that the solver selected then a stable state, which gave the GFP response that the table wanted, but not necessarily what you would see in vivo. So actually, this took about 4 or 5 months of work to develop what we are calling the attribution constraints, which basically directly address the problem of bistable orthogonal designs by preventing a particular gene from being able to influence its own expression, as well as requiring the change in GFP state to be somehow linked to our input signals. So this addressed the problem of BODs, and I just wanted to mention it because this was about 5 months of work, after my comprehensive examination. So that's one of the big advantages that Eugene-SID and Eugene-SIM brings to the table, is that other tools are not explicitly screening for these type of designs. Another advantage of Eugene-SID and Eugene-SIM is that we're explicitly linking the design and modeling steps of synthetic biology applications. So a recent review basically said there's 5 steps: conceptualization, design, modeling, construction, and probing, testing, and validation. We can see that in silico tools are generally used for the design and modeling stages. And several in silico tools have been developed, including Cello, the work of Zamarotti and Muratis in 2014, also their off-circuit work, and a modeling tool like Equip has also been developed. However, none of these tools have explicit link to either modeling or design companion tools. So the workflow is incomplete even in silico. So by creating our Eugene-SID and Eugene-SIM tools, we're able to make that connection. Other advantages that we have include increased granularity, complexity, and detail in our model system. And we're hoping that these two tools together could potentially form part of a synthetic biology application pipeline in future. So we applied this to several different logic tables conceptualizations. So I'll just show you and walk you through how we reported sort of the bulk results or the holistic results. So for this AND circuit that's going to be responsive to cadmium and copper, we're going to list our number of solutions. So in these particular runs, we limited ourselves to 7 days maximum, or a maximum of 1,000 solutions. We're also going to report on the percent of solutions that are proven to be globally optimal. This will sort of serve as a rule of thumb for how well Eugene-SID found a solution or how quality those solutions are. Size indicates the number of triads in a particular solution. So we can see on this number line a minimum, a maximum, and the blue will denote the mode. And then finally, time will also be reported here, including minimum, maximum, and this black bar representing the mean. And then we basically had 27 different circuit conceptualizations that we worked with in order to sort of see how Eugene-SID behaves over a large number of solutions. So here is an example of 9 of those particular conceptualizations. So this includes AND, non-implication, and converse non-implication, which is the same as non-implication, but the arguments are reversed. And we can see in these particular cases that we're going to have a very high level of optimality. We're also going to have sort of a very low threshold for the smallest design, as well as we're going to have fairly quick solution time on the order of less than 100 seconds per solution. And this sort of fits with what we can see in the literature, because we'll commonly see and as well as not imply circuits in synthetic biology literature. On the other hand, when we look at more complex circuit designs, such as half adders, not and exclusive or, we see that we have a much lower optimality ratio, as well as much larger circuit designs and much longer solve times. So this showed that the particular bioparts library wasn't able to let us form these particular logics. But we feel that this is a issue with the bioparts library rather than an issue with the tools itself, because of some of the solutions on this particular slide. So we can see that for a complex circuit, like a NOR circuit, we can have a very high optimality ratio for cadmium and copper logic tables and solution concepts. But once we start adding zinc in as a desired ligand or a desired signal, we can see that our optimality ratio drops off very significantly. So our conclusion from this is that basically Eugene Sim can find these particular solutions. However, it's somewhat limited by the bioparts library that you feed it as to how good these solutions are going to be. So if we have a larger bioparts library, we should be able to model more and more complex circuits more easily. So now that we've looked at sort of the general and high level results of Eugene SID, let's look at some of these specific solutions, these conceptualizations, and see how Eugene Sim works in. So as I mentioned before, our conceptualizations are going to be defined as a logic table. In this case, a cadmium copper responsive OR circuit. Eugene Sim will then design a particular set of triads, which includes promoters, transcripts, and terminators that will give us this desired logic. And then Eugene Sim will model this logic over a period of time. So this particular circuit, we can see that Eugene Sim will give us sort of an OR behavior. And so we're seeing the correct dynamic behavior. And I wanted to point out that this was solution number 41, because what happens in Eugene SID is that we will solve, first try to solve for size 1, then move to size 2 and size 3. So this is going to be an iterative increase in solution size when the solution space for a particular size becomes null, essentially. So this is actually the first solution for size 4 triads from this CD-CU OR conceptualization. And this is the particular circuit design which gave us the strongest results in terms of on versus off. So this is also going to highlight that our first solution, our most obvious solution, or our easiest to construct solution, is not necessarily going to be the best. And so this highlights the usefulness of tools like Eugene SID and Eugene SID for having stronger synthetic biology responses. Another particular successful solution we had was for a copper zinc circuit. This is going to be solution number 11. So this is what a not imply gate looks like. So it's basically going to say if you have copper, have GFP expression, so long as you don't also have zinc. So that's what a non-implication circuit design is. This particular design had 3 triads and showed successful dynamic behavior, although it had a rather long startup period. Now, part of the point of Eugene Sim is being able to screen our solution designs. So in this particular case, we have a cadmium zinc not and responsive circuit that we wanted to design. And for solution number 26, we saw that we had a 3 triad solution and that the dynamic behavior was not what we wanted it to be. So you can see when we have just cadmium presence, we're going to want to see high GFP expression. However, we see that after about time point 5 here, GFP expression will fall off. So this is one particular solution that we would screen using our Eugene Sim results. I did also want to mention here why we didn't sort of combine these two tools into one. This is because a sort of combined Eugene Sim tool was too computationally expensive. We just couldn't get it to run in a reasonable amount of time. So we figured we'd split the two and have a faster runtime, and we figured that would be more useful for users. We also wanted to look at Eugene Sim as potentially a standalone dynamic modeling tool for genetic circuits. So to do this, we created a what's called a repressilator. A repressilator is a synthetic biology design which has sinusoidal behavior and turning on and off reporters in a sinusoidal pattern. And so when we applied our Eugene Sim tool to this, we were able to see periodic behavior as well as able to model sort of complex phenomena like startup behavior. And we were able to keep a very strong and consistent period over time. However, unlike what we might expect to see in vivo, we had more of a wave crest shape to our predicted behavior. This is because of the underlying mixed integer linear programming approach, which we took. And so when we have sort of sharp breaks in our graph like this is because a binary variable which indicates sort of on or off of a controlling enzyme is switching states. So that switch in states is causing this sharp break. Now, again, as with before, we could make this a more discrete on-off tool. However, this would vastly increase our computational costs and decrease our computational tractability, as well as decrease our optimality ratio. So we decided to stick with our MILP formulation. As I noted before, part of the point of this tool is to hopefully be part of a synthetic biology development workflow. To this end, we have created a protocol paper which steps users through how to use both of these tools either together or separately, as well as how to parallelize this for a large number of potential conceptualizations. And I think this is representative of my approach to science, because as a tool builder, I'm very much pro other people using my tools, and I try as hard as possible to make my science reproducible and accessible, as well as to encourage other people to use it. So that's sort of the end of my work here at UNL, but I will be a researcher going forward, and as you might have seen from some of these presentation points, my work isn't quite yet finished, so I'll be passing it on to future grad students. The work that I think is most, has the most potential or the most future promise is my work related to Exophyella dermatitidis. So quick summary is we found that the key enzyme in melanin production, which is called tyrosinase, is well conserved, as well as certain active sites related to albinism are also well conserved. And it has the potential to be used as a model of a human melanocytes. This did leave us with two overarching research questions, though, that can be investigated by our in vivo collaborator. This includes whether or not Exophyella engages in some type of pigment profile optimization, as well as whether or not there is a novel role for carotenoids in the organism, which we have not yet discovered. So this is currently the focus of some ongoing research in the Harris lab at Iowa State, and they're testing this with various panels of mutant Exophyella. I see a couple of future potential research directions for this particular study. The first is that if we can compare Exophyella and human melanin metabolism on sort of a good level, and they have decent, and they compare decently, we might be able to use Exophyella as a test system for human medical applications. This would be particularly impactful for melanin deficient diseases like albinism, and this would be most impactful in Africa. So we can see this particular girl here had her limb removed simply because she was albino. So it causes certain medical and social dangers that we could hopefully treat in future. Also, independent of how well the human angle works, we can study Exophyella as a model polyextremotolerant organism. And we can use our knowledge of synthetic biology to redesign particular organisms for the extremotolerant trait using these pigments in order to adapt them for post-climate change in extraterrestrial environments, potentially. So this is sort of why I feel ExoPhylla has some of the most potential of my research projects. Now, to this point, I've been talking exclusively about one type of genome scale model. However, there is a second. So the one I've been talking about to this point are called stoichiometric models, or SMs. And we can build these from genomic knowledge. They use linear programming, and they're accurate for some applications. However, they're limited in that they are linear because this makes them a bit too simple to model some complex and dynamic behaviors that might be in metabolism. We can also really don't include regulation in a very robust manner when we use these models, and generally they're modeled at steady states. The other type of genome scale model which could be produced are kinetic models of metabolism. These have several advantages, including having greater accuracy, incorporating regulation, and having more sort of enzymatic and reaction scale detail. However, they're limited in that because they are nonlinear, they're much less computationally tractable, as well as they are more difficult to produce. So like I said, they're more computationally expensive. We also need to have more knowledge of the system ahead of time in order to build these particular models. So one example I'd like to give is the largest stoichiometric model that I've seen is for humans and contains about 10,000 reactions. The largest particular kinetic model that I've seen is for E. coli, and that contains less than 500. So there's about a 2 order of magnitude scale difference between the two. There are many modeling approaches and tools which are currently there for kinetic models, which try to limit these particular disadvantages. 2 of the ones that I will be using the most in my future research will be ensemble modeling in an algorithm called KFIT, both of which try to predict kinetic parameters and or kinetic regulation. My particular future as a researcher will be as a postdoctoral scholar at Penn State in the Marani's research group, where I will largely be studying kinetic modeling of cyanobacteria, probably for some kind of desired bioproduction, as well as tool development for kinetic models of metabolism, sort of scale up my genome scale model tools for kinetic models so that they're useful in other applications. At this point, I would like to thank everybody who's helped me throughout my PhD journey, particularly the members of the Systems and Synthetic Biology Laboratory. These would particularly include my advisor, Dr. Saha, who sort of gave the dramatic changing of the labs in December of 2017, and without him, I most definitely would not be standing here right now. I would also like to thank one of my fellow graduate students, Mohammed Mazharul Islam, who is a first author co-author on one of my papers, as well as just for his advice over the years. And our new graduate student, Niaz Bahar Chowdhury, who is sort of taking over some of my projects, and I have worked with on getting a few papers out as well. I would also like to particularly thank the undergraduate students who have worked with me here at my time at UNL. This includes Josh Bauman and Connor Lund, but most particularly, I would like to thank Anna Maber for all her hard work in the Eugene Sid and Eugene Sim project. She put in a lot of time and is an author on both the journal and the protocol paper. I would also like to thank the high school researchers that have worked with me for the past couple of summers, particularly Molly and Evan, as well as Ali, Kareem, and Bree, who also worked on the Eugene Sid and Eugene Sim tools. And I would finally like to thank my funding sources. I started by being funded by my advisor's faculty startup grant from UNL. And I am now funded by the Center for Root and Rhizobiome Innovation here at UNL. And since this was kind of a whirlwind tour of what I've been doing as a PhD student, you can follow this QR code, which will link to my Google Scholar profile if you want to read my papers, or because of the nature of conferences for the past 2 years, some of my conference presentations are available on this particular YouTube playlist. And if you're interested in other work our group does, you can check out our group YouTube channel or follow us on Twitter to keep abreast of all our updates. And with that, I would like to thank you for your time and attention, and would be happy to take any questions you may have. Oh, and for those online, just to type the questions into chat, or just say that you have a question into chat. That way I can mute us and we don't get feedback when you ask your question.
Or anyone online?
I'm not seeing any activity in the chat yet, but let's see if anybody's got a raised hand here. No, I don't see any raised hands. So, Mohammed, yes, thank you.
Thank you for the very nice talk. Could you explain for the OptFill project, you had to run OptFill multiple times,
Yes.
right? So in stages.
Yes.
Could you explain why?
So I ran them in stages because of the different confidences I had in our databases. So let's see how quickly I can scroll back up here. So I had 3 different databases that I was working with, built from 4 different models of Aspergillus. And basically, the first database I had the most confidence in, because that was going to be in 3 of 4 Aspergillus models. So these blue stars. So I felt these blue stars, you know, if I added them in to fill a gap, I felt reasonably sure that I could use various tools like BLAST in order to identify it in the genome of Exophyella. However, with these particular databases, say the one of, you know, where a function was only present in one of the models, I was less confident that these particular functions could be found in Exophyella. Therefore, I wanted to put them at the end of my OptFilling procedure. So I had 3 applications of OptFill because I had sort of 3 different levels of confidence of my database.
What do you think is like an interesting implication of the fact that you found that there's more water being excreted because of metabolism as opposed to transport?
Can you repeat the question?
Yeah. So what Adil asked is, what do you think is an interesting implication of the results from the DFBA work about finding that water was a very significant contributor to metabolic, to transpiration? It's a very good question. I haven't thought too much about that. I would say probably it means that we need to rethink our current understanding of water requirements for plants, and know that you know, the water required for plants, especially as droughts continue to intensify throughout the world, and especially the Western United States. I think we need to recognize that some of that water is going to be produced by the plant, so that we can maybe better optimize the water that we give to our farmers in our farm fields in order to sort of minimize our waste of water, especially as water becomes a precious commodity.
Why, first of all, was very excellent presentation. To be honest, I still didn't wrap my head around it, right? But you mentioned something about using it Africa, right?
Yes.
So I would like for you to shed more light on that.
So let me go to a particular couple of slides. So albinism in Africa, there's 2 dominant kinds. The first is oculocutaneous albinism type 1, which is the type where there's a mutation in tyrosinase. And this mutation in tyrosinase basically causes it to be non-functional, and therefore they can't produce melanin. However, let me hide this bar real quick. There we go. So, however, there are other types of melanin. So that's oculocutaneous albinism type 1. That's in Caucasians and Africans. Oculocutaneous albinism type 2 is actually a bit more common in Africa. It accounts for about 70% of albinism cases. And this is where there is a transporter protein located on these organelles called melanosomes that actually transports tyrosine, which is the precursor to melanin production, into the melanosomes where the tyrosinase is located. So if there's that mutation, then it just can't get to the tyrosinase. So that's sort of the second kind of albinism in African populations. And we think exophylla could also help us model that because exophylla, the melanin synthesis really occurs in the cell wall. So tyrosinase gets pumped out into the cell wall. So we're essentially hoping to treat the cell wall and what happens here as semi-equivalent to what happens in a human melanocyte in order to try to address these two types of albinism. There are other kinds of albinisms, and these vary by race. So for instance, oculocutaneous albinism type 3 is a mutation in tyrosinase-related protein, which also shows high homology with exophyllous tyrosinases. So this would this most affects South African populations, and this would allow us sort of a third avenue of investigation of albinism. And oculocutaneous albinism type 4 is a mutation in generally found in Japanese populations in a melanocyte located protein, but they haven't really nailed down what its function is yet or why it causes albinism. And then there are many other more increasingly rare types of albinism. There's all the way up to oculocutaneous albinism type 7. So what we're hoping to do is basically use our knowledge of the proteins and the tyrosinase in exophylla and our ability to study it in the lab with a much simpler genome and proteome than humans have in order to figure out if there's some kind of drug which might be able to restore a mutated tyrosinase to function. Yes. Okay, from Dr. Belander, we have a question. Hi, Wheaton. Can your model report on oxidative phosphorylation efficiency for any one cell type? Energy yield from burning a molecule of glucose is a very important stress test for the impact of the synthesis of a recombinant protein. Short answer is yes, we can model that. We can figure out how much energy we produce from a single molecule of glucose or a single unit of glucose being fed to a cell. So yes, we can. Practically, we have not yet because that has not been a focus of our research, but that is certainly another research direction that we can take.
Anything else?
No, it was just that.
Any other questions? All right.
So it's about Eugene, Sid, and Sim. So for that, just thinking of like how you would apply this to multiple different organisms.
Yeah.
What size of library would you think you would need for this to be useful?
Right. So the current library that I used for the data that I showed had about I think it was 24 promoters, about 7 or 8 transcripts, about 7 or 8 enzymes that corresponded to those transcripts. So that's a fairly small database. At least I would think it's fairly small. It's actually a lot bigger than other databases that have been used. But to be useful, it really depends on what kind of conceptualizations you're working with as to how big of a database you need to be useful. So if you're looking at these basic logic gates like an AND and an OR, you can do that with a database of 4 or 5 promoters, 2 or 3 enzymes, 2 or 3 transcripts. But if you want to make a more complex logic gate, say if you want to build in an adder, you're probably going to need double or triple that to be useful. So like I said, it's really going to be variable on how complex your conceptualization is.
Any other questions?
We have another on chat. So Dr. Keshwani says, great job, Wheaton. You've had a unique opportunity to understand biological models of complex biosystems. Given your experience, what do you wish the general public knew or understood about biological models? Ooh, that is an excellent question. Okay, so your biological model is only going to be as good as the other researchers doing the actual wet lab experiments, publishing their work. So for a biological model to work, we need well-funded science sort of across the board, both within experimental labs and in vitro labs and in silico labs, so that we can really collaborate. So a biological model is sort of built upon the shoulders of giants, if you want to borrow Newton's particular quote there. So we're just using other people's data, other people's research, and we're grateful to them. And we're really dependent on the open and free exchange of science in order to build our models and encourage other people to use them. So we're real big proponents of open science and being usable and transparent.
All right, no other questions. Let's thank Wheaton one more time. And thanks all of you guys.